I really appreciate everybody who's joined us here today. We see opportunities to really provide tools that will allow us to support COVID-19 research in various ways. And in this case, I think, certainly in looking at the application of high throughput genomics for sequencing of the SARS-CoV-2 virus. So to talk through the outline here today, the talk is really going to focus on sequencing approaches to SARS-CoV-2, and I'm going to give you a little bit of background into our approach on that. Part of that will be introducing the plexWell technology that we have built our approach on, and how we've integrated that with the ARTIC network amplicon protocol that was created to support multiplexed PCR and sequencing of different type of viral samples. From there, we'll go through a SARS-CoV-2 sequencing case study that we built internally here, and we'll talk about some of the performance features of that we think will be interesting for labs around the world who are interested in sequencing coronavirus samples. So at seqWell, we really focus on making library prep tools that can help accelerate genomic applications and across a range of different problems. We think of library prep really as effectively the software of the genomics era, a critical tool for really matching the capabilities of what sequencers can do to the research and clinical problems that people and researchers are looking to solve. Many of those applications, as I'm sure many of you are aware, really have a lot of common challenges that they are encountered. For example, workflow, scalability, and getting better data out at the back end. And really, we think of library prep as being the key tool for solving some of those challenges, and thinking about ways of creating library prep tools that will improve the performance of sequencing by creating, for example, more integrated, better workflows that are simpler to execute, can process more samples, and give better information out at the end. As you heard a little bit from Joanne, our history and partnership with Adgen is certainly part of how we have built some of those tools. Going back a few years, we started working with Adgen really in how to, as Joanne mentioned, create a robust and scalable NGS-based plasmid QC and validation pipeline. Certainly part of that was based on, what is really a critical issue is that in order to do that with NGS and convert from what was previously a Sanger-based pipeline, we really had to address kind of some of the real key issues there. For example, large variations in plasmid content, and the fact that most assembly algorithms do require very tightly and well-controlled libraries in order to get reproducible performance. So we really built a solution based on our tunable plexWell platform, which was really optimized to solve that problem, and has been used now for many thousands of plasmids in the Adgen repository. As we, as many other researchers and companies, awoke to the realization a few months ago, certainly the challenge of COVID-19 now was certainly apparent to us, and the urgency became clear in February and March. And we started to look at ways that we could also help in that area. And certainly looking at genomic tools and NGS multiplexing being at the core of many of the challenges that researchers are facing. And we started to look at opportunities really to address some of the challenges for sequencing that were emerging. Sequencing of SARS-CoV-2 really has a lot of clear impact, for example, in understanding and responding to different types of epidemiological questions. Certainly the tracking of patterns of mutations, identifying modes of spread, and looking at the overall trajectory of the virus as it spreads around the world. It's really important to realize that sequencing is also has a distinct utility that's really different from the diagnostic testing, which is of course critically important. But sequencing provides really an important layer of information in terms of the actual mutations of virus, and being able to track those mutations and connect patterns of spread around the world. And we've certainly seen, I'm sure as many of you have in the media, numerous reports where we can make certain assessments and conclusions about how the virus continues to spread. So we, in thinking about that problem, we certainly were aware of the need for multiplex sequencing. And so, we really felt that our plexWell platform could be a really ideal platform for us to build a more scalable sequencing based approach. Just to say a little bit about plexWell and what it is, plexWell is a multiplex library prep platform that we created that really allows you to take large numbers of samples and process them in parallel to make a single library off of multiple samples at the same time. One of the really key attributes of this technology is that it allows you to take input samples that have a range of different concentrations and input amounts, and pull them together after a simple tagging step and produce a normalized library out at the end. Really, this is an effective way of processing, many types of samples where you are interested in multiplexing. And of course, it's really key to getting scalable sequencing. So the workflow is really designed to allow you to do, for example, 96 well plate assays in a matter of a couple of hours, but really also scale up to, in many cases, hundreds or thousands of samples. And this is an example, of our workflow for a 96 well plate, where, as you can see, we take 96 samples and can pull them into a single tube and process that certainly very quickly and with potentially multiple plates at the same time. What this really allows is for what we call scalable multiplexing. The idea behind this is that, again, you can replace what would normally be a library prep process where you're taking individual samples and making individual libraries, and replace that with a process where you can take multiple samples and make a single library off of them at the same time. One of the real features of this, again, is the normalization component, which really saves a lot of time compared to conventional library prep workflows. As you can imagine, there's a range of different applications where this approach is very useful, and we've certainly seen successful application of this in areas such as low-pass whole genome sequencing, single-cell RNA-seq, and as Joanne mentioned, amplicon and plasmid sequencing. Of course, sequencing of viral specimens is certainly well connected to these types of applications, and we approach that from the point of view of having a multiplex PCR foundation, as you'll see in the next few slides. But the key takeaway here is that plexWell is really a streamlined way of making multiplex libraries very simply and efficiently with improved overall time, cost, and performance. So in thinking about this problem, we were aware of efforts by researchers to create sequencing tools for the SARS CoV-2 virus. And most notably, a group known as the ARTIC network had created a protocol for doing multiplexed RT and PCR that essentially allowed researchers to take samples and process them through the RT and PCR steps to the beginning point of library prep. And we felt that was a really ideal starting place for potentially applying plexWell. First of all, because the ARTIC protocol, being somewhat well established and used by researchers around the world, was familiar and certainly has been optimized and is something that people are now widely using. So we really felt that this was an excellent place to start, and of course the potential here is again to be able to take that process and plug it into a library prep workflow, which would allow for sequencing of hundreds or thousands of samples at the same time. To say a little bit about the ARTIC network and what it is, it's a consortium of laboratories that have been created. The history goes back to certainly early work by Josh Quick and Nick Lohman on the Ebola virus and Zika virus a few years ago. They have taken that same approach and built it into a sort of a widely used platform for doing multiplexed RT and PCR. Certainly in the last few months, as labs have awoken to the challenge of how to collect information from coronavirus specimens, this has been one of the more commonly used platforms. And so, for example, there have been several publications, a couple of which I note here, that have used this in different library prep workflows to sequence samples from different types of collections. The typical ARTIC protocol really is again taking the multiplex PCR and combining it with a downstream library prep with different workflows. The most commonly used workflows, again, that had been reported before were generally either ligation-based workflows where one takes the PCR products and simply puts them through a ligation-based library prep. Of course, this comes along with all of the requisite normalization steps. Alternately, certainly in a similar vein, there is a Nextera XT, sometimes miniaturized protocol as well. Again, both of these are really designed to allow individual ARTIC samples to be processed and turned into libraries that can be pooled and sequenced. You know, our goal here was, again, to take and evaluate the potential of taking the ARTIC protocol and using it as part of plexWell in order to really enhance and really accelerate the speed with which multiple samples could be multiplexed in a single assay in, you know, for example, a few hours. So in looking at this problem, one of the first things we really had to assess was the working range that we would be seeking to achieve. And just to go through a little bit of the background on this, certainly in looking at you know, the data coming out of RT-PCR, the qPCR sort of diagnostic assays, typically, you know, it's been reported that, you know, the average viral load in positive samples is in the sort of average in mid-20s, 24 or so CT range. What this really means is, again, our goal was to allow labs that were implementing testing pipelines, we wanted to provide them with a protocol that would allow typical positive samples to be plugged into a sequencing workflow downstream to essentially act as a sequencing method that could be deployed on positive samples. And so in looking at that, we really assessed the working range that was necessary. And for example, in looking at that, we established that really the working range is generally between about 1,000 up to, in some cases, many thousands of copies of virus per sample. And so really our goal then became, okay, can we establish the sequencing performance for a working range that goes down to 1,000 copies of viral RNA into the assay? And that became sort of the baseline goal for our approach. So this is an overview of the protocol that we have then put together based on looking, again, taking the ARTIC protocol and making some slight modifications and combining it with plexWell. I'll note, as you see in the lower right here, that we have made this protocol available on our website. You're welcome to go and review that and read it in more detail. But the key features here really are, again, showing, you know, and again, you'll see data in the next few slides on examples where we can take sample human nucleic acid samples with spiked in controls variously from either the twist or ATCC references, input that as the beginning for the RT and multiplex PCR part of the ARTIC protocol, and then without actually needing any purification, go directly into plexWell and make the multiplex libraries that can be loaded directly onto sequencers. So the next few slides, I'll be presenting data that sort of go through the results that you achieve using this protocol. So one of the first things we really were interested in looking at here was, can we achieve, you know, high coverage libraries that that are sufficiently well covered even with low viral copy number. What you can see here is again a range of viral copy numbers going into the RT and multiplex PCR, and then put into plexWell, and you know that the amount of data you get back certainly tracks with the amount of virus that goes in, but certainly down to a reasonable limit of 800 copies or so. We have sufficiently high high coverage to say that we're likely going to be able to confidently call variants in those viral genomes, and that's really borne out in this next plot that I'm showing, where you see the percentage of the actual SARS CoV-2 genome that's covered at different coverage levels, 1x, 10x, and 30x, depending on the number of viral copies that went into the RT and multiplex PCR. So again, this gives again another view of the data, which shows that above a number of viral copies, essentially in the mid-hundreds, certainly to 1,000. Above 1,000, we have uniformly high coverage of samples going into the assay. And again, as that sort of, you know, again, needs to play out, of course, is how does that translate into variant calls? And what you can see here is the sensitivity, with essentially our ability to detect the known variants in those samples. For example, there are 3 variants that are present in the ATCC reference versus the original Wuhan strain. And our ability to detect those and accurately call them both positively and negatively also tracks very well with coverage. So certainly up to, again, a threshold of 800 copies, we have 100% sensitivity and specificity for those variants. Again, the uniformity of performance, again, in that working range, again, our goal here was to be able to really see that we were getting predictable and uniform performance across a wide range of viral copy number. And as you can see here, ranging from as many as 100,000 copies down to 800, you see, again, uniformly high coverage across the full SARS-CoV-2 genome and mapping rates that are well above 80%. So this certainly gives you an illustration of the performance that can be achieved here, combining the ARTIC protocol with plexWell. Of course, the other advantage that really plexWell brings to this problem is the ability to scale it up. And so what we then looked at is really a sort of larger scale study where we were going to take a large number of samples and multiplex this to make sure that we got uniform performance in a much larger multiplex context. So in this case, what we did is we took a single input plate with a range, again, of different viral input amounts, again, across our full working range, and made 4 replicate 96-plex libraries, which you see illustrated in terms of their raw read counts here. And then, again, looking here at the ability to cover each of those genomes at some minimal level that would be required to call variants. And the performance there, again, was quite good, especially if you factor in, again, several orders of magnitude difference between the low and high amount of viral RNA going into the assay. The actual amount of data coming out at the end was actually quite good and uniform. So this really is, again, a good example of how one could scale this assay up. And as I'll mention in the next couple of slides, we do have plexWell assay configurations that can support up to 1,152 samples in the same run. So just to say a few words in summary here about the ARTIC and plexWell protocol, and again, our work here. Certainly the key findings from this work is that we can achieve robust performance, looking at the protocol being able to make high quality libraries in the background of human nucleic acid, which would be needed for full genome sequencing of the coronavirus genome. Certainly an improved and simplified workflow compared to other methods without the need, for example, of normalization or purification of the amplicons going into library prep. We feel like this is a key feature that will allow this protocol to be deployed more quickly and easily in labs. And again, establishing a working range which is matched to the likely number of viral RNA genome equivalents that would be going into this assay in the context where it's run on samples that were tested positive via the sort of standard RT-qPCR assays. So again, that working range is certainly supports that you're going to be able to get high quality genome coverage down to the sort of low end of what RNA copy numbers of positive samples would generally be. And of course, one of the primary benefits of plexWell that certainly applies here as well is the ability to take, again, a simple workflow and scale it up. So again, regardless of what type of sequencer you might have in the lab, whether it be a MiSeq or a NextSeq or even a larger sequencer, we have different kit configurations that can support, up to 96 samples per run, or multiples of 96 or 384 samples per run. So again, it gives that flexibility in terms of the number of samples that can be loaded onto the sequencer and run in your lab. So in conclusion, really, I think the advantages of this method are robust performance, simple workflow, and the high multiplexing capabilities. I should also note this is because of the fact that we're saving, for example, in certain parts of the workflow that would otherwise be needed, such as normalization. This is a very cost-effective solution. Being able to make a single library from many samples takes a lot of steps out of what would otherwise be usually a more difficult library prep workflow. And so this really is going to translate into significant cost savings overall versus many other methods. It's also automation friendly. Again, we certainly don't require automation to use the plexWell process, but it certainly is friendly to automation. And in fact, we're currently collaborating with Opentrons on an open source protocol here to implement this and distribute it to researchers around the world. And it would certainly be easily amenable to automation on other platforms. And we have experience seeing plexWell automated on Biomech and TCAM platforms, for example. In terms of future directions and other areas we're working, I will just say a few notes about this. Yeah, certainly in terms of the multiplexed RT and PCR process, I think there's certainly some improvements that we're very interested in exploring there and are working on. For example, simplifying the workflow, improving the performance and limit of detection of the assay, and really saving some of the time that's associated with that part of the workflow. And of course, in the scalability side of things and being able to really have this assay perform, at larger scale and even better, we are looking at additional barcoding index reagents and looking even at the ability to support unique dual indexing as part of this protocol in order to really get the best performance for a range of different assay needs. So with that, I'll conclude here. Again, I appreciate everybody joining us here, and I'm certainly happy to take questions. I'll just put notes here that if you had any questions about the protocol or certainly the workflow that we presented here, I certainly invite you to visit our application support page at seqWell.com. And of course, if you want to learn more about the tools that Adgen has created, adgen.org will have all the information that you'd be looking for as well. So thank you very much. So if there are any questions, I will certainly be happy to take those at this time. So one of the questions that came in on Q&A here, and I'll read through a couple of these. There's a question here regarding the spike in control from ATCC or twist. So what we have done here, the question is it spiked into the sample? Is it a limit of a blank control? So we took the ATCC and twist samples and tested both of them as spike-ins to a total nucleic acid purified sample at different, essentially different titrations to assess the performance of the assay for different amounts of virus that would be in a typically encountered scenario there. Next question I see here is how this is different from COVID-seq that was just announced by Illumina a couple of days ago. So, as you may be aware, the COVID-seq assay is a diagnostic assay that is meant to, in a way, sort of replace or allow a more scalable form of the RT, what would otherwise be done with RT-qPCR. It doesn't actually sequence the full genome, so it's really designed as a quantitative assay to really assess the specific amount of virus that's in a sample, but it doesn't actually give you the full sequence information of that virus. Here's a question for, I think, let's see, we have a question here. How can you increase the sensitivity and sequencing and pooling of the workflow? Have you seen false negatives? So because this is really not designed as a diagnostic assay, our focus has really been on achieving good sequencing performance in a range of RNA amounts that would be coming in, in samples that would have typically been tested positive, kind of upstream. It's a good question, though. I mean, again, I think as you see again with the use of sequencing as a sort of do everything assay, if you can improve the sensitivity of the assay in order to be able to detect lower and lower amounts of virus, then certainly including quantitative controls, I think certainly would be a good way of being able to, you know, make this a more quantitative assay. But our focus right now has been really on getting uniformity of sequencing coverage, so that, you know, one can use this to get, you know, reproducible, high quality genomes from samples that you have sort of independently tested to actually contain virus. Another question here, can we elaborate on customers who are currently using plexWell for COVID-related sequencing? And does it work for custom amplicon panels or fewer than 96 samples? So certainly I can say a few things about that without you know, sharing information from some of our that would be, you know, kind of more confidential. But I think, you know, we do have some of our users who are using this for different types of amplicon sequencing workflows in the COVID application space. Again, it ranges from, you know, sort of different sizes of amplicons and different numbers of amplicons in that assay. In terms of custom amplicon pools and panels, absolutely. In fact, I think, you know, I think this is a real great area for exploration here is, you know, coming up with different panels and different content that can be used as part of this protocol. And then as to whether it can be used on smaller numbers of samples, absolutely. We actually have a kit called plexWell Plus, which is really designed to handle, you know, lower multiplexing batches from between 8 and 96 samples. I have a question here from Fritz Roth. Hello, Fritz. Fritz says, thanks, Joanne and Joe, for your efforts on SARS CoV-2 plasmid sharing and genome sequencing. Question is, it seems like one of the major advantages of plexWell beyond indices is skipping normalization. Can we say more about how that works? So yeah, I'm happy to do that. If we go back to our workflow, I can actually share the slides here. It will be easier to talk through this live. The key to this process really is this iterative barcoding procedure, taking sample barcodes and tagging samples in the first step with a equal amount of sort of different barcoded reagents in a 96-well plate. What that does is it effectively tags those samples and creates the same number of tagging instances that once those samples are pooled, allows you to make a library in which, as long as you've got the same number of tags in each of those samples, you're actually tolerant to different amounts of DNA. So it's that sort of limitedness of the sample barcoding step combined with pooling and making a library off of that pool, which effectively gives you that normalization performance. Question here from Randy Kerstetter. Hello, Randy. He asked, would we comment on the analysis that is done at seqWell? So I'm going to infer from this is this maybe I'm going to interpret from this he probably is referring more to the plasmid analysis pipeline that Joanne mentioned earlier, and not the sort of standard bioinformatics pipelines that we've been running on the COVID data that we've been generating. We've created a very tuned algorithm that's really designed to take advantage of some of the features of plexWell libraries in terms of the expected size and diversity of the libraries, so that we have optimized the protocol with Adgen to use on plasmid libraries. And I think that tuning and combined with, again, you know, the tight control of the libraries has been the key there. Without getting too far into the weeds on the actual steps of the analysis, it uses sort of standard, you know, De Bruijn graph type assemblers, using, you know, different pre-processing steps to make sure that we don't put too many kind of noise-containing reads into the assembly. And then we do a remapping process after assemblies are made in order to go back and make sure that we can get confident read calls throughout a fully assembled plasmid. Final question I see here from one of our attendees is just the cost per sample. So first of all, I will say, as it relates to the work in COVID, we are actually, again, if you go to our website, there is a promotion that we're doing here to really enable researchers who are trying to solve different types of problems in the sequencing of COVID samples or SARS CoV-2 genomics. I think that's really the first place I would direct you. In general, our kits are priced very competitively and really designed to allow you to hopefully process more samples, saving essentially saving more per library. And certainly, I would encourage you to reach out and follow up with one of the members of our great commercial team to discuss that more. One of the other questions here is again, mentions template switching can be an issue for applications with multiplex libraries, where barcodes can be connected to other barcodes in a library. Do we have diagnostics in our analysis pipeline that can detect and remove template switching events that can otherwise cause mismapping between libraries? Yeah, so this is a great question, and in fact, I think there's a couple of detailed issues there that that certainly would require in-depth discussion. In general, what we find for most mapping applications, again, the rate of template switching that we observe in these libraries, which are again large complex libraries that have many barcodes present, is far below the amount that would generally be introduced in any kind of standard mapping or variant calling pipeline. And by controlling the amount of that switching below a certain level, which was part of the optimization process of how we have created these kits, we certainly are confident that that is not a problem that is going to cause false variants, for example, to be detected. And again, it can also be approached as a sort of threshold setting problem, where you simply have to know the amount of that template switching and essentially remove or ignore any reads that might be observed at or below the expected level, which in our typical typical assays that we've looked at is below 1% in any library. So again, that's a succinct answer to a somewhat complicated question that I'm sure, if you reach out, we would be happy to talk about more. So I think there's, I don't see any other questions here. If we, maybe we could conclude here. I think we're running close to our allotted time. But certainly, I think we really appreciate everybody's attendance here today, and it was great to have the ability to talk about the work we're doing, and certainly hear about the work Adgen is doing to support the distribution of tools for a variety of applications, including COVID-19 research.